Just grouping organisms according to their similarity, their structures, and then behavior based like that. It is a very wide branch of biology, and it is so important because each organism has to be identified. If a biologist is communicating about an organism, it should be so specific, and that is what we call classification. Now, we group these organisms in what we call taxa, or taxonomic groups, according to their relationship with each other. We call those taxonomic groups, or you can just call them taxa. That's how we're going to group them. So basically that is it. So in the levels of classification, how do we do the classification? How does the classification occur? We have a grouping system that we use to classify these things, and majorly we will start with the highest to the lowest. That means at the higher level we have many numbers. We have a large number. We have many, and then at the lowest level we have low numbers or few. Let's say few. So at the beginning we have a kingdom as the highest. We have a kingdom as the highest level of classification. As we have increase in numbers, that means here you are going to have both man and say a cow and say maybe a snake. But then by the time we reach down here, you discover that we have narrowed it down so that you are only in the same species with people or with organisms that resemble you. Organisms that resemble each other will belong here, and actually this is a level where organisms can even interbreed, viable interbreeding to produce fertile offspring. But any part up this side, there is no interbreeding. The different kingdoms that are in that we actually have. But majorly we are going to concentrate on this animal kingdom. Now what you're going to look at today is in kingdom Animalia. That is how we call the kingdom. It is not scientific to say kingdom animal or animals, to say kingdom Animalia. And they have, or they possess these characteristics that you see right here. We know that these are multicellular organisms, and their cells do not have a cell wall. In plants, plants are multicellular, but their cells have cell wall. But these are multicellular with no cell walls in their cells. And then they are diploid throughout their life with haploid gametes. Now what happens is that diploid is a double state. A haploid state of the gametes is a half state. That means that at fertilization, a haploid and a haploid will have to meet to form a diploid organism throughout life. Gametes, haploid, then again when they meet in fertilization, they form a diploid zygote. So they are heterotrophic. This means that they do not make their own food. They feed on already manufactured food in whatever way they do it, according to the way you will see it in nutrition. And then another thing is that zygote is formed after fertilization. And this zygote usually develops into a hollow ball that we call the blastula. After fertilization, a zygote formed. Remember haploid, forming a diploid zygote. That zygote multiplies or divides into a blastula, which is a hollow ball called the blastula. Now, in the Animalia as a kingdom, we have two groupings. We have the Vertebrata and the Invertebrata. Now, what happens in the Vertebrata, which we have the Vertebrates, what you call Vertebrates, we have the fish. This is like tilapia, Nile perch. We have birds. This is like a hen at home. We have the amphibians like the toads, the frogs. We have the reptiles like the lizards, the snakes. We have the mammals like the cow, goat, the man. And then we also have the Invertebrata, which are the invertebrates. Here we have the protozoa like the Amoeba, the Paramecium. We have the nematoda like the hookworms. We have the platyhelminths like the flatworms. We have the Annelida like the earthworms. We have the mollusca, like the snails. We have the arthropoda, like the spiders and the bees, the insects. We have the coleotrata and the chidomanta. Here we have the starfish. Now, let's look at what composes the invertebrates. These are the invertebrata, okay? We have the first phylum, which is the Phylum Protozoa. This is a very important phylum in biological studies, because it is here that you have the euglena, the paramecium, and then the amoeba. Looking at the characteristics of this, they are single-celled organisms. That's why we say they are unicellular. Another thing is that they are microscopic. Just the fact that they are single-celled organisms tells us that they are microscopic. All of them, you have to use a microscope to view them. You can't see them with your naked eye. Then they reproduce by means of binary cell division or cell fission. That means they can divide. One cell can divide into two cells or can divide into many cells. Binary means two. So one cell dividing into two cells is called binary division, and then cell fission can be one cell dividing into more cells. Fission, okay? Then movement is in paramecium by cilia. The hairy structures that surround the paramecium are called cilia. They keep beating as they provide a swimming movement. And then euglena uses the flagella. That is just one tail-like organ that keeps flapping and the euglena moves forward. And then the amoeba uses the pseudopodia. These are projections formed from the cell membrane of the amoeba. Now let's look at the characteristics of the amoeba, just the amoeba only. We said that amoeba uses the cell membrane to engulf food. This is a process we call phagocytosis. Usually, the food is the cell membrane of the amoeba invaginates and surrounds the food, and when it encloses the food, a food vacuole is formed, and then the food can be digested, and then the waste particles can be released later. So this one is called phagocytosis. And then in locomotion, we still use, the amoeba still uses the pseudopodia. What happens is that the projections form on the cell membrane due to the flowing of the cytoplasm inside the amoeba, and then it moves forward. And then we have excretion is by diffusion through cell membrane. Any excess thing, any ammonia or waste product formed always diffuses outside through the cell membrane by what we call diffusion. That is depending on the concentration gradient. If there is more waste in the inside than the outside, they'll always move to the outside by diffusion, as you'll see in transport. And then excess water is always pumped out by structures we call contractile vacuoles. As water is taken into the amoeba, inside the amoeba by osmosis, it forms contractile vacuoles, collects in the contractile vacuoles, and then they are released by the contractile vacuoles fusing with the cell membrane. It is a continuous process. We said they reproduce by binary cell division. One amoeba divides to form two daughter cells. What are the economic importances of the amoeba? We have amoeba feeding on bacteria. Now, if amoeba feed on bacteria, and we know that bacteria causes diseases, that means that the amoeba helps us in controlling disease. And then we have Entamoeba histolytica. You should note that this is a scientific name. That's why I've written it like this. Entamoeba histolytica causes amoebic dysentery, which is also a disease of